Okay, the projector is working now, so let's start. Is the volume high enough? Good. All right. So in this lecture, we're moving downstream from the receptors. The last lecture, we talked about the basic properties of receptors, and today we're going to start from the receptors and going to a specific type of receptors, and then moving downstream from the receptors and going through the first signal transduction pathway of this semester. So before I start, let me tell you a story of the cholera epidemics. This is truly the world's first global disease. The first major epidemics actually happened in the 1800s. Actually, the outbreak starts in 1817 from India. So we know that the cholera toxin has been festering in the upper Ganges probably for centuries, but most of the time it's restricted to the local area. In 1817, during early part of the year, the Great Kumbh's Festival happened in India, and during that 3 months festival, people shared waters. They all bathed in the Ganges River and shared open camps along the banks of the river. So at the end of that festival, people from lower Ganges or from other part of India went home, brought the cholera toxin with them. And this is the disease that's spread it's a waterborne disease. If anybody get in contact with the feces or waters that has been contaminated by the feces from the patient, they will get disease, and it happened really fast. So the disease started here. This is the map of the 1800s. This is where Calcutta is. That's where the first major outbreak actually occurs. And then when people went back home to different parts of India, they brought the cholera disease with them. And that's not all. There are people who start trading along the ancient trading route to Iran, Baku, and along the Volga to the inner part of Russia. And by that time, the cholera epidemic starts spreading. And in Russia, in the mid-autumn, there's a big festival, merchant fairs, actually. And at the end of that fair, the merchant went home, brought home with them the cholera epidemics to different part of Europe and different part of Russia. Eventually, this epidemics aided by the start of the Industrial Revolution, steam-powered boat, ships, trains, brought the cholera from port to port, ships to ships. And during that time, the economy started happening, but the quality of life is still the same. And in many parts of the world, in many big cities, you have big slums where people live together, and the sewage, there's no such thing as a closed sewage underground. A lot of times, the sewers are open systems where the things go underground and leaks everywhere. So the cholera epidemics moves really fast. And by 1831, it hit England. And from England, that was the time when Industrial Revolution is happening, and people really focused on making money and having the economy going, and they don't want to quarantine ships because that will really damage the business. So, and then during that same period of time, Ireland is suffering from the major potato famine, and there's a lot of people try to flee that poverty, so they get on the ship and travel to where? America. So those ships, of course, are also in very bad conditions. People who the owners of the ship want to make money. So in a ship that's designed for 150 people, they put in 500 in it. And of course, there's no good sewage system. Everything go outside, overflowed. So by the time the ship hit New York and Montreal, cholera epidemic hit those towns as well. So in New York, the doctors realized it's a very horrible disease because cholera is the type of disease that within 12 to 48 hours can kill a person. And the record from the time says that you can have a person who's totally lively and energetic walk on the street in the morning, and by midnight is dead. And the cholera, when it hit, the patients suffer from high fevers, severe diarrhea, and extreme dehydration, and died really, really quickly. So New York almost become a ghost town at the time. The medical officials want to start quarantining whole cities or whole towns. And certain personnel, who else? A banker always stood out at a critical time and asked the physicians, have you considered what the impact of your action will be on the city's business? And because of that, they didn't do the quarantine. And of course, the disease spread everywhere. So bankers always do the right thing at the right time. So this is during the 1832. That's when the first cholera epidemics hit New York, and then also gradually spread out to other parts of the world, Latin Americans. If any of you have read Gabi Marquez's novel, Love at the Time of Cholera, that was talking about the cholera epidemics that eventually hit Latin America. And that's the time when they start to reintroduce the sanitation system to Latin Americans. It's actually very interesting. Now, during the first cholera epidemics in England, there's an eminent doctor, Dr. John Snow. He first discovered that he noticed that actually the cholera epidemics, the cases of cholera, seems to have a link with contaminated water source. But nobody wanted to believe him. And so the major weakness of his discovery is that he could not pinpoint what is that poisoned water that's causing the disease. And so the whole medical profession essentially ignored him. And by the end of the 1830s, the first cholera epidemics died down, and the people have this mentality that out of sight, out of mind. They don't want to do anything. They don't want to think about anything. 20 years later, 1854, the second cholera epidemics hit London again, hit England. And according to history, this is the worst cholera toxin ever outbreak ever occurred in England. So this time, Dr. Snow had a chance to further gather data and then prove his point. So he had this very strong case where he found that within a period of 10 days, there are 500 cases of cholera epidemics occurred all within a radius of 250 yards, centered around the famous London Broad Street pump. And this is one of the public water pumps where everybody used water from there. So everybody who touched that water pump, share the same water source, get disease. There are only two exceptions, and that's one pub and one school where they have their own private wells that's not contaminated water. And nobody, not a single person from those two places ever gets sick. So it's a very strong case that the disease is linked to the contaminated water. And it was based on this discovery that the government official finally start this whole field of public health where they introduced modern sanitation and the sewage system that spread all over the world. So, but he's still not able to pinpoint what causes the cholera disease. And it was not until 1876, a German doctor, Dr. Robert Koch, first identified it's actually a bacteria that's named the Vibrio cholerae as the cause of the disease. However, people don't understand how this bacteria actually caused such a massive disease. It takes about another 100 years until about the time when you guys start to be born, 1980s, that finally people figured out how does cholera toxin actually kill people. And the target of this toxin is a G protein. And so with this beginning, we start today's signal transduction pathway. So where does a G protein function? The major part of cell surface receptor that G protein function is called the G protein coupled receptors, or GPCRs. There's another name called 7 transmembrane receptor family. And the reason is that this receptor family, the G protein coupled receptors, all shared this structure. It's one big polypeptide that going in and out of membrane 7 times. So that's why we call it 7 transmembrane receptors. It's not just one transmembrane domain, but 7 of them go through from the end to the beginning to the end. The N amino terminus of the molecule is right here outside, and the C terminus of the molecule is inside. So this is the part, the outside is the part that binds to ligand, and the inside is the part that G protein interact. So the molecular characterization have shown that G proteins can interact with, have at least 2 contact with both of these domains. It's a big chunky protein that contact this domain. And so when you have ligand binding here, you might have conformational change that allows the G protein to bind. So this receptor is the largest receptor family that's found in human. and in nature in general. It's one of the most common receptor families in eukaryotic site, and it's very conserved during evolution. Even the most simple eukaryote, like yeast, contains seven membrane proteins, receptors. For example, yeast use this type of receptors to mediate mating. So the mating factor receptors are actually a G protein couple receptors. And in human, it covers many aspects of physiological function, from adrenaline hormone responses, to smell, vision, and odor. In mouse, you have about 1,000 different CRs just to mediate different taste and smells. So mouse is very, very sensitive to the taste. Humans, the number is slightly less, about 700 of them, which is pretty large anyways. So this is very large families, and there are a lot of ligands, different type of ligand that combine to this family of receptors, going from small molecules to proteins. And often, pharmaceutical industry also target these receptors because it's also involved in many human diseases such as cancer. Thyroid hormones receptor is one of the GPCRs, and a person without a thyroid hormone receptor would have a problem with development and also mental retardation, and also get thyroid tumors. So the pharmaceutical industry also developed drugs to target different part of this receptor, and it's a very druggable target because it's huge. But we actually don't know everything we need to know about the receptors because most of the receptors are in the membranes. It's very difficult to study a membrane protein. If you want to do a crystallographer to look at the structure of a protein, this is one of the ultimate challenge of that structure study, so we don't know enough. Okay. So the signal transduction pathways of GPCRs, the first set of steps have three major components. Okay, so this is the membrane, the gray part. This is the receptor we're talking about. In the absence of any ligand, sometimes they saw the ligand, they call it hormones. It's the same thing. In the absence of any ligand or hormones, the inactive receptor sit on the membrane, doesn't do anything. Now, on the other part of the membrane, there's trimeric G proteins. Remember the trimeric G proteins we were talking about last time? It's called trimeric because it has three subunits. It has alpha subunit, which is the big one here, that bind to GDP in the absence of ligand. So this is in the inactive state. The alpha subunit bind to GDP, and also the alpha subunit interact with the two smaller beta and gamma subunits. All three subunits are tethered to the plasma membrane through this little links. So it is also a membrane protein, okay? And there's another component that's very crucial for signal transduction, and that's called the effector protein. Now, I'll gradually tell you what are the individual effector proteins are. So this is the generic name. In the case of adrenaline, epinephrine receptor, it's adenylyl cyclase. So those are the three components. Now, in the absence of ligand, receptors in the inactive form, the G proteins are in the GDP-bound form, and this effectors also in the inactive form. What happens is when you add ligand, we'll go through, this is a very busy slide, and the next page shows a description of what we're going to talk about. So I'll go through slowly. This is the part that we just saw, the inactive state. Receptors, GDP-bound trimeric G proteins, and inactive effectors. The first step in the process is that the ligand combine to the receptor, and that causes a conformational change. And so in this picture, it shows the color changed. It's actually conformational change. Now, when the conformational changes, this whole receptors act as a GEF, a GTP, guanine nucleotide exchange factors. Remember that the G protein, when it goes from a GDP-bound form to GTP-bound form, sometimes they need the help of GEF. So the active receptor, in a way, it kind of functions like a GEF, because now the conformational change does two things. Number one, it exposes the domain now that the G protein can actually physically bind to it. So you see it's shown here, all right? And when it binds to it, it functions as a GEF to kick out the GDP. So that binding in a way, also cause the conformational change in the G-alpha subunit. And then G-alpha subunit change conformation, leave out the GDP. Now the space here is open. And so by chance, whatever is in the cells will come to bind to this pocket. Inside the cells, the concentration of GTP is much higher than the concentration of GDP. So when you have an empty space, by chance, what comes to bind is GTP, because that's much more abundant. All right? So when GTP start to bind to G-alpha, that causes another conformational change, which now G-alpha leaves G-beta gamma. So this has been the traditional view, but recently the new study found that the G-beta gamma does not have to be physically separated from G-alpha, but they can do their own thing. So there is a conformational change. Some of them dissociate, but some of them don't. So that's one of the things. The reason I mentioned this is that I want you to know that a lot of the stuff I'm telling you is a work in progress. What you learn today that's correct, maybe 10 years down the road, or maybe 5 months down the road, has been proved to be wrong. So just remember that. Don't be surprised. We understand very little about cells, and so we are teaching you what we understand. But maybe your own work will prove they're wrong in the end. So G-alpha subunit, once it binds to GTP, it becomes active. Okay, so this is a guy that's able to transmit the signal downstream. And the way it's doing that is now it's in a conformation that it can bind to this effector molecule. So you can imagine as it expose the site of interaction so that the effector protein can bind. So this GTP bound form is the active form. Now once it binds to the effector molecule becomes active. It changes conformation. At the same time, the effector molecule also functions like a gap. A GTPase activating enzyme or activating protein that accelerate the ability of G-alpha to hydrolyze GTP to return it to the GDP bound form. Okay, so how long does G-alpha exist in GTP bound form determines how long the signal, the active signal, can keep going. So normally this whole thing happened really fast. This G-alpha subunit bound to GTP only would exist in a matter of seconds and no more than minutes. So it's a minute to second level. It's very short. So you see the whole thing then gradually return to its state. So the receptor, if you look at the receptor, going to the green, the active form. Now once the GTP is bound, the G-alpha subunit dissociate from the beta gamma and both dissociate from the receptors. So the receptor now can return to the inactive state, ready for the next round of signaling. The G proteins going from GDP state, change conformation going to GTP, and then hydrolyze it, go back to the beginning state. The effector molecules bind to GTP-bound G-alpha, become activated, and eventually moves downstream signal transduction. And eventually will go back to the end state as well. So the key point of this whole thing is that the GTP-bound form of G-alpha has to exist. And this is the key point in signal transduction. And what cholera toxin does is to actually modify this protein through adding a ADP ribose group on it. And this process essentially fixes G-alpha in the GTP-bound form. It can no longer hydrolyze GTP. And so it stays in this form indefinitely. And so in the patient that actually have this GS molecule, it stayed in GTP-bound form indefinitely, and this pathway is always active. And a lot of the receptors of this family actually exist in the intestine cells. And the function of those intestine cells is to essentially pump the chloride, iron, water out of the cells. And so when you have this pathway constantly active, and the cells obviously is always pumping the chloride, iron, and water out of the intestine. And on the broad scale, we call that diarrhea. And so that's how it kills the patient. Okay? So the cholera toxin is actually very smart. It picked the most important target and keep it in one stroke, keep it in the most active state forever. And essentially whatever happened downstream doesn't really matter that much. or whether you have ligand or not doesn't matter that much. The signal is constantly on. Okay? So when you study signal transduction, we often take advantage of these different drugs or toxins. And that's how we understand the importance of these turnovers. We can, for example, use this chlorotoxin in the laboratory to essentially fix the G protein in this form, and we ask what are the proteins that's now being turned on without even ligand. So these are different ways to manipulate different steps in signal transduction, and that's the part of the logical concept thinking that I'm talking about. So this is just essentially going through what I said to you, so I won't read through to you again. So how many G proteins are there? There are many, many different types. And what's listed here is only a partial list. So the ones that we talk about with the epinephrine receptor or beta adrenergic receptor is what we call the alpha, a G alpha S. S stands for stimulatory G protein. So the stimulatory G protein essentially activate the signal transduction pathways. The effectors that it uses is called adenylyl cyclase. And this enzyme functions to generate the second messenger, that's cyclic AMP. Okay, and we're going to talk a little bit more about that. Now, on top of stimulatory G proteins, we also have inhibitory G proteins. And no matter which type of G protein is talking about, they all have the alpha, beta, gamma subunit structure. But the sequence is different, and they do different functions. In the inhibitory G protein families, they can also target adenylyl cyclase. But instead of activating it, it actually inhibits its activity. So you have a decrease in cyclic AMP activity. It also regulates ion channels directly. There's olfactory G proteins that we're going to talk about in the next lecture that essentially is responsible for mediating all the different smells that you can feel. This is also a trimeric G protein. It also activates adenylyl cyclase. So you see that many of the G proteins, even though the individual receptor is different, G protein is different, they use the same effector proteins and generate the same second messengers. There's and some of those G proteins activate a different effector molecules called phospholipase C that we're going to talk about next lecture. And phospholipase C then mediate the generation of different second messengers. There's also G alpha T, stands for transducing, that's responsible for changing the activity of cyclin GMP phosphodiesterase, which decrease cyclin GMP concentration. And that's responsible for our vision, that when we see things, it's going through this G protein. So these are the major classes of G proteins, and I don't need you to really memorize the details. And usually, if I give an exam, I'm going to say that this is the pathway that use the G alpha olfactory, which usually activate adenylyl cyclase, and then I'll ask you other questions. So these are you don't need to memorize this whole table, okay? But I hope that it sounds pretty familiar to you. So this family has many, many members, and even G olfactory has many different isoforms because we smell different odors. And every odor uses a different G alpha olfactory. Okay, so it's a large family of proteins. Now, G beta gamma are not totally useless. They can also mediate some of their own functions. And I'm not going to discuss any of those for this class. So, as we said, the effector protein for the stimulatory G alpha protein is adenylyl cyclase. So this, again, as I said, is a transmembrane protein. We know that it sits on the membrane. The function of this protein is to use ATP as a substrate and modify it to become cyclic AMP. And the chemical reaction is shown here. Again, you don't need to memorize the structure of this thing. This is not a chemistry class. This is ATP. You see the 3 phosphate groups. And when you have adenylyl cyclase, 2 of the phosphate go away and the other one circle around. So that makes cyclic AMP. So this enzyme has this catalytic activity, which is to catalyze this chemical reaction. And this enzyme can be regulated both in a positive manner, as we said, through G alpha S, the stimulatory G protein, when it's bound to GTP, right? But it can also be inhibited by the inhibitory G alpha protein when that is bound to GTP. So all these trimeric G proteins share the similar idea of activation, that they need to bind to GTP in order to be activated. Now, once they're activated, the different proteins, the activation definition is different. For GS, the activation definition is to activate this protein. For GI, the inhibitory G protein, when it's active, it inhibits this guy. Make sense? So it's doing its wrong function, but it all has to be in the GTP-bound form when it can really activate the effector protein. So it shows you that there are different proteins that regulate this pathway. For example, the ligand here is the epinephrine, the so-called adrenaline, glucagon, ACTH, and there are also proteins that inhibit this pathway. We call the inhibitory hormones that bind to their own receptors, which go through inhibitory G alpha and then inhibit this pathway. Okay. So that gets to the first part of the signal transduction. You go through receptor, go to G protein and adenylyl cyclase, resulting in the generation of cyclic AMP. And this is the second messenger. Now, in the cells, normally without any ligand, cyclic AMP is existing at around the concentration of 10 to the minus 7 molar. So it's a very low concentration molecule. The reason that it's kept low is because whenever you have adenylyl cyclase that activate this process, there's another enzyme that get rid of that. That's called cyclic AMP phosphodiesterase, which is essentially convert cyclic AMP into AMP. And this thing happened also pretty fast. And in the cells, you have the balance of this activity versus that activity to keep cyclic AMP at the constant balance equilibrium. And when you have outside signal that activate more adenyl cyclase, then of course this part of the reaction overwhelms the bottom part of the reaction. Then you get an increase in cyclic AMP concentration in the cells for a very short period of time, because one of the downstream consequences of cyclic AMP activation is to, again, later activate this enzyme to get rid of it. So all of these activations happen very fast and also only last a short duration, so that your body does not have to go through adrenaline rush all the time. Okay. All right, so this is just a brief summary of the first part of the signal transduction pathway we talk about. This is the general form, and this is the specific ones that we talked about. So you can have ligand bind to a specific G protein coupled receptor, in this case, adrenaline bind to beta-adrenergic receptor, and that activate a trimeric G protein. In this case, it's G stimulatory G protein. And then that results in the activation of effector protein. In this case, is adenylyl cyclase. And that generates the second messenger. So the goal of this three-member signal transduction pathway is to generate the second messenger. And then what we're going to do now is to say, what does the second messenger do? Now remember, all these three proteins sit on the plasma membrane. So when they transmit signals, of course, they can't go very far. But when they generate a small second messenger, life is different. The second messenger can diffuse into the cytosol, into other part of the cell really fast. And that's why you want to do this. And there's another benefit that we're going to talk about slightly later, and that is signal amplification. Okay? So a small number of ligands can generate relatively larger number of second messengers. And what does the second messenger do? Now, the second messenger also have a target. And in this case, the target is a protein kinase called pKa. Now, cyclic AMP activate cyclic AMP-dependent protein kinase. And this protein kinase is also abbreviated as pKa. So whenever we talk about pKa, you should know it's cyclic AMP-dependent protein kinase. So this protein kinase has 4 subunits. Without cyclic AMP, it's in the inactive form, because the 2 catalytic subunits stand for C here are usually bound by the regulatory subunit, which essentially inhibit its kinase activity. So without ligand, this whole complex is in the inactive form, the inactive pKa, meaning its catalytic subunit cannot do anything, cannot induce phosphorylation because it's inhibited by the regulatory subunit. Now, when you have a lot of cyclic AMP, cyclic AMP start to bind to the regulatory subunit, and that changes the conformation, allows the catalytic subunit to dissociate from the regulatory subunit. And once free, the catalytic activity, which is a protein serine threonine kinase activity, become activated. And though these guys can go out and then really phosphorylate its downstream substrate. And this has many, many different substrates. It's one of the most important or abundant protein kinases in the cells. So you see the signal transduction cascade. We already covered all three major general modes of signal transduction that we discussed in the last lecture. You have ligand binding that activate a G protein, and through G protein, we generate a second messenger, and from there, we activate a protein kinase, which allows the post-translational modification of many proteins. And then we can see what type of physiological response we're getting. So once the PK is activated, it can phosphorylate substrate. Now, pKa exists in every cell type in your body. And every cell type has a different set of pK substrate. So you will get a different physiological reactions, actually, depending on which substrate it's phosphorylated. Now, we said generally when the proteins are phosphorylated, there's usually two things that can happen. One thing is you can change the enzymatic activity of that protein through this post-translational modification. Or you can also activate gene expression to change the abundance of that protein in the cells. So we're going to mostly talk about this fast change because this involves new transcription, new translation, and that takes time. But the fast response is happening through changing the enzymatic activity of a protein. When we have adrenaline rush, it happens right away. You can't wait for hours or days for it to happen, right? So this is really the most important part. Now, in terms of epinephrine, which is adrenaline, when you have adrenaline rush, we know the feeling, right? Every part of your organ seems to change. So you can see that the PK substrate exists in multiple cell types. It happens in liver, so that the idea of adrenaline rush is so that you get more energy. So energy comes from where in human? Glucose, exactly. So the whole process, no matter how different they are, is to increase the production of glucose so that you have more energy so you can run away from danger. So in the liver, it increases the conversion of the glucose precursor glycogen to glucose. And when you increase the hydrolysis of glycogen to glucose, you want to inhibit the opposite process, which is glycogen synthesis. Glycogen synthesis happens using glucose to generate glycogen. You want to inhibit that because you always want to go the other way. And you want to increase synthesis of glucose from amino acid, from lipid, from any source of glucose, right? And then in the muscle, you have to, the heart muscle, cardiac muscle, you want to increase the contraction rate so that you get more blood into the limbs so you can run away. Adipose tissue, you have increase in hydrolysis of triglyceride, again, to generate more glucose, to give you energy. And the intestine, increase in fluid secretion. And many people experience that when you're really nervous, either go on running or going to an important test, you want to go to the bathroom a lot more often. That's what happens. Your adrenaline is doing it. When it's very severe, of course, the cholera, in the case of cholera toxin, then you have severe diarrhea. So in the next two slides, I'm going to show you some examples of how pKa really coordinate this whole process of generating more glucose. So this is at high cyclic AMP condition, which is when you have a lot of hormone running, beta-epinephrine running in your body. When you have the adrenaline rush, you want to have increased glucose production. So at this point, high cyclic AMP would activate a lot of pKa, right? So the pKa would then do two things. One is it stimulates the breakdown of glycogen to glucose and also inhibit the synthesis of glycogen from glucose. These are the two opposite processes, and you don't want to waste energy, and therefore you coordinate both. How does it coordinate? All through the process of phosphorylation. So the enzyme that regulate glycogen hydrolysis to glucose is this enzyme GP, glycogen phosphorylase. The enzyme is inactive unless it's phosphorylated. So once it's phosphorylated, it becomes active, and then that hydrolyzes this process. Now, what phosphorylate? Glucose phosphorylase. I think that you've learned all that in MCB 100, right? Anyway, so once this phosphorylation is controlled by glycogen phosphorylase kinase, and that kinase is only active when it's phosphorylated. And what controls the phosphorylation of this kinase? pKa. Okay, so it activates a kinase cascade. pKa activates a kinase cascade to induce this process. Now, for the inhibition, it can directly phosphorylate glycogen synthesis, and that's enough to inhibit its activity. So that blocks it. Now, there's another interesting phosphatase. That's called the phosphoprotein phosphatase. So whenever you have kinase that phosphorylate things, you need to have phosphatase that take it off. And so all of these phosphorylation events can be taken off by this phosphatase called the phosphoprotein phosphatase. This phosphoprotein phosphatase, therefore, if you want to keep these phosphorylation events happening, you need to inhibit that phosphatase. And the inhibition is through this inhibitor protein, inhibitor of phosphatase. Now, this inhibitory protein can only bind to the phosphatase once it's phosphorylated. And that phosphorylation is achieved, again, by pKa. So it's pretty much one protein coordinates the whole event, increase glucose production, inhibit glucose synthesis, and also inhibit the opposite process, the phosphatase that takes this whole thing off. Now, when the cyclic AMP level goes down, the whole thing reverses. And the reason it reverses is because this phosphatase. The phosphatase becomes now active because pKa, when the cyclic AMP level goes down, pKa returns to the inactive state. Nothing maintains the phosphorylation or inhibition of this phosphatase, then this guy becomes active. So the phosphatase in the cells normally are in the active state. So they comes in, dephosphorylate everything, and then that reverse the process, increase glycogen synthesis and decrease glucose production. So this is how the whole thing gets coordinated, and this is only the process in liver, and we haven't even dealt with the adipose tissues and all the other things. So things can be very, very complicated. So that's the example shown here, and every tissue has a whole set of pKa substrate, but the detailed process is different, but the principles are very similar, okay? So the great thing is this whole process, you can see there's a significant amplification of signals. Now, when we have adrenaline rush, we all know that how profound this whole effect is. Is it because that the adrenaline is produced at such a high level that we are all flooded with adrenaline? And the answer is not really, because if you look at the concentration of epinephrine in our blood, at the peak of adrenaline rush, it's around 10 to the minus 10 molar. That's 100 picomoles. That's very, very low. And yet the effect that we feel is very profound in every tissue. And the reason that we can achieve that is because the signal transduction cascade that really amplifies the signal. So you can start with 10 to the minus 10 moles of molar of epinephrine. Each epinephrine binds to a receptor. Now, each receptor, GPCR, can activate about 100 molecules of trimeric G proteins. So you have a signal amplification there. Now, each G protein activates one adenylate cyclase. And each adenylate cyclase can generate 100 cyclic AMP molecules because that's a catalytic enzyme. So from here to cyclic AMP, you get a 10,000-fold signal amplification already, okay? So that's a very interesting thing. Now, cyclic AMP, of course, combine to protein kinase pKa and activate many pKa molecules. And pKa being the catalytic enzyme, it can phosphorylate many more substrates. So you get a massive amplification. Not only the number of substrates that get activated, but the type of substrate that get activated. So you get a broadness, a very widespread of the type of response you can have through activating this one key enzyme. So by the time epinephrine get to the physiological response, like how much glucose you generate and that sort of thing, the signal has been amplified 10 to the 8th times, 100 million times. So this is a very powerful system, and that's why we need signal transduction cascade. because every time in the cascade, we achieve two things. One is a small number of signals generate a large amount of response. And number two is that one molecule can generate many types of responses through the broadness of these individual mediators. And so one epinephrine can trigger response in liver, in the adipose tissue, in cardiac muscles, in intestine, all over the place. So all through these critical sets of signals. Okay, so it's a very, very powerful system. Now, if the system is so powerful, it can generate such amazing response, you want to make sure the signal gets turned off really fast. Otherwise, you'll be in permanent adrenaline rush state, and that's really not a very pleasant state. So there are many built-in mechanisms to turn off the whole signaling. Number one, if the signal transduction can be amplified 10 to the 8th time that easily, how do you control it not blowing out of proportion? So one thing it controls is the number of these different molecules in the cells. They're all limited. You don't have unlimited number of receptors. You don't have unlimited number of trimeric G proteins. The number of molecules for all of these are controlled at very strict number in the cells. And then during the process of signal transduction, you see that every time when you go to the next step, the previous step get returned to the native state right away. So, for example, the affinity of receptor for its ligand, the first step is decreased whenever the G protein is bound to GTP. So that's saying, okay, the signal is going downstream. That's enough. I don't need to bind to ligand that well. So that sort of blunt out the first step of the signal transduction pathway to really mellow down the response a little bit. The second is the intrinsic GTPase activity of the G alpha S is enhanced when it binds to the next step inhibitor, the effector molecule. As I said, adenylyl cyclase is functioning like a GAP, a GTPase activating protein. It makes this process go much faster. What does it mean, this process go faster? When you have GTP bound form, this enzyme becomes activated at hydrolyzed GDP, return it to the GDP bound inactive form. So once it activates the next step makes sure the first step go back to its native state as fast as possible. Okay, so that's what it is. Phosphorylation of different substrate by pKa is transient. This is very important because pKa functions in so many different cell types, phosphorylates so many proteins. If you don't turn it off right away, they will keep going. So they can be, a lot of these proteins can be rapidly dephosphorylated by protein phosphatases. As I said, without signal, this phosphatase activity is maintained at a certain level in the cells. It's active. Okay, and you need to inhibit that in order for the adrenaline signal to go on. And without that, this protein become activated again. That keeps all the phosphoprotein level low in the cells. And again, cyclic AMP, as I said, is a very short-lived second messenger. It's being produced by adenylyl cyclase, but there is this phosphodiesterase enzyme that return it to the ATP, AMP form to terminate its response. So this molecule, even though it's very powerful, can bind to pK, it does not exist there for a very long time because this enzyme will be activated by pKa, again, through phosphorylation, which come back and hydrolyze it quickly. So pretty soon, the whole system returns to the baseline, and that's where after your adrenaline rush, you start to feel your leg becomes soft and you can't even stand, and all of these previous things quickly go away. So that's because all the system resets. And that's very, very important. So this is just a final summary of the pathway we talk about today. It's GPCR activation that activate a trimeric G protein. In this case, we talk about stimulatory G proteins, which activate adenylyl cyclase, generate cyclic AMP to activate pKa, and that results in changes in glucose metabolism, in gene expression. So in the next lecture, we're going to again talk about the same pathway, but with a different effector enzyme and different second messenger.